So let's just very quickly remind ourselves of the vasculature of the kidney. So the large vessels come in and they become the interlobar vessels here in the medulla and end up eventually as the interlobular vessels out here in the cortex. And these vessels end up as little clumps known as glomeruli, or a singular is glomerulus, and we have about 1 million of these in each kidney. So this is the structure of the nephron. You can see the glomerulus in this little sac here in the space known as the Bowman's capsule. The filtrate goes through the proximal convoluted tubule, comes down into the medulla and becomes a loop of Henle, goes up, becomes a distal convoluted tubule, and then eventually ends up as a collecting system which produces urine. I want you to note that the capillaries in the glomerulus come out as an efferent arteriole, which become vasorectal, which run parallel with our tubules, and it comes all the way down here as little hairpin capillaries that come that are parallel to the loop of Henle, and these are known as the vasorectal, which literally means straight vessels, and they supply blood and oxygen to the medulla. So for this video we're going to be focusing on the loop of Henle and what's known as the countercurrent multiplier. And the countercurrent multiplier, this is just the process of pumping out salt into the extracellular fluid as a mechanism of concentrating our urine. So fluid comes in from the proximal convoluted tubule at a concentration of about 300 millimosmoles per litre. Now this thin descending loop has many special channels known as aquaporins, which allows water to leave the tubule. So this is just water and fluid going out. So as water leaves, so it's quite not as concentrated out here, but as water leaves, you can imagine the fluid inside the loop of Henle becomes even more concentrated as we descend, as water is leaving. Now this thick ascending loop, the ascending loop, is impermeable to water, which means it hasn't got these aquaporin channels to let water out. But what it does do is it actively transports sodium chloride, or in other words salt, outside of its tubule. And what this does is it makes the extracellular space more salty. It increases the concentration of the extracellular space. So that means it gets very concentrated down here because water is leaving and it stays concentrated. But as these concentrated solute, the sodium chloride, gets pumped out, it becomes more dilute as we go up again. Now, because it's concentrated in this extracellular space around here, this allows a gradient for water to pass through passively from the descending loop of Henle. Okay? I'll say that again. Because the concentration in the extracellular fluid is increased because of the ions being pumped out from the thick ascending loop, it allows a gradient for water to leave passively in the thin descending loop. So this quite nicely sets up the countercurrent multiplier system in the loop of Henle. So as we can see, because water is leaving the thin ascending loop, because of the highly concentrated extracellular space, this allows the fluid inside of the tubules to become even more concentrated. And as it ascends in the thick ascending loop, which is only permeable to ions, these ions, sodium chloride, gets pumped out to essentially make it more concentrated, more salty in this extracellular space, giving a means for water to passively move out. You might notice here how urea is also actively transported out of the collecting tubule to further concentrate the medulla. Finally, remember how we said the loop of Henle is actually covered by these vasorectal capillaries? Well, you would expect that because these capillaries are running through, it would sort of wash out all the solutes in the loop of Henle and basically make it less concentrated here and make this whole countercurrent mechanism useless. But actually, because the vasorecta are highly permeable to ions and water, it maintains an equilibrium with the extracellular fluid, so it doesn't wash out the solutes. So it basically continues supplying oxygen to the medulla without washing out the solutes and ruining the concentration of the extracellular gradient. And this in itself is known as the countercurrent exchange mechanism. All right.